Hey everyone, I'm Dr. Indira. Today the topic is glycolysis. We have already discussed the glycolysis, the software that is required for glycolysis, silent features of glycolysis, and initial pathway that are involved in a glycolysis. We have discussed all this portion in a glycolysis details part 1. So if you think you missed those part, then you can please go and check it out in my profile. It's already there. So let's get started with the glycolysis part 2. Okay, so we have already started glycolysis pathway. Glycolysis pathway consists of energy investment phase, splitting phase, and energy generation phase. Okay, so we have already studied the energy generation phase, energy investment phase, where the energy has been invested in the form of ATP. ATP is consumed in the initial steps of the glycolysis. We have consumed 2 ATP. First ATP is consumed in a first step of glycolysis. After that, phosphorylation of glucose, there is isomerization reaction to form fructose 6-phosphate. After isomerization, again another phosphate has been introduced, so and again another ATP has been consumed. So eventually 2 ATP, total of 2 ATP has been consumed in the energy investment phase. So we obtain fructose 1,6-bis phosphate. After that, this fructose 1,6-bis phosphate is equally splitted into a 3-carbon compound, glyceroldehyde 3-phosphate and dihydroxyacetone phosphate, by the help of enzyme fructose 1,6-bis phosphate aldolase. And these two components try to maintain the equilibrium number, so they are interconvertible to each other, and this interconversion is carried out by the help of enzyme triose phosphate isomerase. Okay, so now we are starting with the energy generation phase of glycolysis. So this glyceroldehyde 3-phosphate is actually involved in a glycolysis. Okay, so why, I will tell you in step 6 of the glycolysis. So now glyceroldehyde 3-phosphate is involved or consumed in the glycolysis, because glycolysis is all about generation of ATP. So now this glyceroldehyde 3-phosphate will produce NADH. It gets oxidized to produce NADH, and during the process of oxidation, it also involves the inorganic phosphate present in the cytosol. So this inorganic phosphate is placed at the first position, first carbon of glyceroldehyde 3-phosphate. So initially it only had phosphate at the third position of carbon. Now it will have phosphate at its first position as well. So the phosphate is present in a first carbon and a third carbon. So that's why the name of product is 1,3-bis phosphoglycerate, which is also a high energy compound. Okay, for 1,3-bis phosphoglycerate releases around minus 49.3 kilojoule per mole of free energy. Okay, so that's why this glyceroldehyde 3-phosphate is producing NADH, which will enter into the mitochondria through malate aspartate shuttle, and it will participate in electron transport chain, and through oxidative phosphorylation it will produce around 3 ATP as per the old calculation, whereas according to the new calculation it will produce around 2.5 of ATP. So during the oxidation of glyceroldehyde 3-phosphate, it is also involving inorganic phosphate in its first carbon of glyceroldehyde 3-phosphate. So this reaction is carried out by the enzyme glyceroldehyde 3-phosphate dehydrogenase. Since oxidation reaction is occurring, so dehydrogenase involving in glyceroldehyde 3-phosphate, so the name of enzyme is glyceroldehyde 3-phosphate dehydrogenase. Now let's move on to the dihydroxyphosphate as well. Okay, so in dihydroxyacetone phosphate, this is actually involved in the formation of important intermediate that is required to form a triacyl glycerol. Okay, triacyl glycerol. So this glycerol dihydroxyacetone phosphate phosphate will produce a glycerol 3 phosphate. Okay, so this glycerol 3 phosphate combines with fatty acid to form a triacylglycerol, and the name of enzyme involved is glycerol phosphate dehydrogenase. And next thing is, it is not giving away NADH to us, it is actually utilizing NADH plus AS plus, meaning it is reducing dihydroxyacetone phosphate to glycerol 3 phosphate. Okay, so NADH is not being made available by dihydroxyacetone phosphate, which is required to produce an energy through oxidative phosphorylation. Okay, so now another importance of this step is this enzyme glyceraldehyde 3 phosphate dehydrogenase can also be inhibited by iodoacetate or arsenate having a valency 5. Okay, it will inhibit this enzyme and might inhibit the glycolysis process. So now let's move on to another step of glycolysis, that is step 7 of glycolysis. So we have already obtained 1,3-bis phosphoglycerate from the step 6 of glycolysis. So this 1,3-bis phosphoglycerate, what it decides now is that it will think that okay, I've already received the phosphate, I have kept that phosphate at a first position of first carbon, so let me just donate that phosphate. So it will decide to donate the phosphate present in a first carbon. So now it becomes 3-bis phosphoglycerate. Okay, so this is 1,3-bis phosphoglycerate name because the phosphate is not present together. Okay, these phosphate are not present together, so these phosphate are present separately, at one at third position of carbon, another in first position of carbon. That's why the name is 1,3-bis phosphoglycerate. If the phosphate would have been present together, then the name would have been one like diphosphate. Okay, so okay, let's concentrate in this section as for now. So now 1,3-bis phosphoglycerate is releasing a phosphate present at a first carbon, so that phosphate will combine with adenosine diphosphate that is already present in the cytosol to form an adenosine triphosphate. So this is one of the example of substrate level phosphorylation. Substrate level phosphorylation means when the substrate itself will release the inorganic phosphate, and that inorganic phosphate will combine with adenosine diphosphate to produce an ATP. That is called a substrate level phosphorylation. So this is a first substrate level phosphorylation occurring in glycolysis in the step 7, and the enzyme involved is phosphoglycerate kinase. So now you must be noticing there is a word kinase. The name of enzyme is ending with the word kinase, and initially we had discussed that there are 3 irreversible steps in glycolysis, and all the enzymes that are involved in this irreversible step of glycolysis are ending with the word kinase. So the name of this enzyme is also ending with the word kinase, but we don't see the irreversible reaction over here. So this is one of the exceptional case. Okay, so phosphoglycerate kinase is an exceptional case where it is not forming an irreversible step of glycolysis. So now let's move on to the another step of glycolysis, that is step 8. Now 3-phosphoglycerate, which we have obtained from the step 7 of glycolysis. So now 3-phosphoglycerate will undergo an isomerization reaction. Now this phosphoglycerate will decide, oh, I'm so bored of placing like phosphate at the same point. Now let me shift this phosphate to the second carbon. So it will shift the phosphate to the second carbon of the phosphoglycerate. So this is this is an isomerization reaction. So the name of enzyme is ending with the word mutase, and this is occurring in phosphoglycerate. So the full name of the enzyme involved is phosphoglycerate kinase. Okay, there is just an isomerization reaction occurring in step 8 of glycolysis. Now let's move on to the step 9 of glycolysis. The 2-phosphoglycerate which we had obtained from the step 8 of glycolysis from the isomerization reaction will undergo formation of enol molecule. So that's why the name of enzyme is also enolase. So how it is forming the enol molecule? So the carbon containing hydroxyl group is forming a double bond with another carbon. So that's how the enol molecule has been formed, and during this process there is a release of water as well. So the O is present over here, and O is present in a third carbon of the phosphoglycerate, and hydrogen present in a second carbon of phosphoglycerate comes together to form water and is released from this reaction. So that is how phosphoenol pyruvate has been formed. Phosphate present at the second carbon. Now another significance of this step of glycolysis is that magnesium is required for the enolase enzyme to act on a substrate, but now this fluoride will combine with magnesium and phosphate to form a complex, and that will bind to the active site of enolase. So when it binds to the active site of enolase becomes like difficult. It won't have a site to attach its software, so it won't be able to attach with the software, so the further glycolysis won't be occurring. So we can use this in our benefit. How? In some condition, when we don't want the glycolysis to occur, then we can use this to our benefit. So in which condition we don't want the glycolysis to occur? So when we have collected a blood sample and it will take time for us to reach the lab, or it will take time for us to run the test, then at that time we need to prevent the glycolysis to occur in a blood sample. Okay, so in that case we require the help of fluoride. So how we will use? Suppose we are going to collect the blood sample from a certain location, and we need to transport that sample of blood to the lab. So during this process there might be a glycolysis occurring in the blood sample, so eventually that will give a false result, meaning it will give a lesser amount of glucose than what it actually would have present in the blood. So we will collect those blood sample in a tube, blood collection tube containing a sodium fluoride. So that sodium fluoride will inhibit the further glycolysis, okay, preventing the glycolysis, so that we can get a actual value of a glucose level in the blood sample when we reach the lab. So another in which condition we want to prevent glycolysis? So you might have noticed some trace amount of fluoride is present in all the toothpaste. So what is the work of this fluoride? Our mouth oral cavity is colonized by an anaerobic bacteria, and the main role of those bacteria is to cause the tooth decay, and it thrives upon the fermented or refined sugar. So in order to get the energy from those sugar those sugar needs to be it needs to undergo the glycolysis process, okay, anaerobic glycolysis. So then the bacteria will thrive upon those sugar. So when we don't let those bacteria to thrive upon those sugar, then we need to prevent the anaerobic glycolysis, and those glycolysis is prevented by the fluoride. That's why the toothpaste that we use consists of some trace amount of fluoride. So in that condition we want to prevent the glycolysis. In two conditions we prevent glycolysis in real life: when we want to run the blood sample for the glucose, or when we don't want anaerobic bacteria to get all the glucose, feed upon all the sugar, and then start to decay our tooth, okay, in that condition. So that was all about the step 9 of the glycolysis. So phosphoenol pyruvate is a high energy compound. So phosphoenol pyruvate releases around minus 61.9 kilojoule per mole of free energy. in the reaction. Okay, so this phosphoenol pyruvate consists of phosphate at the second carbon. So now it also decides to release the phosphate present in the second carbon. So when it releases the phosphate present in second carbon, it will go and combine with the adenosine diphosphate present in the cytosol to form ATP. So this is a next example of substrate level phosphorylation. So in glycolysis, there are two steps involving in substrate level phosphorylation: step 7 and step 10 of a glycolysis. Step 7 consists of phosphoglycerate kinase. Okay, let me say phosphoglycerate. Sorry, not this one. Phosphoglycerate kinase, step 7. Okay, and step 10 is pyruvate kinase. So that was all about the aerobic glycolysis. In case of anaerobic glycolysis, this pyruvate would undergo to form a lactate, okay, by the help of enzyme lactate dehydrogenase. Okay, so now let's move on to the irreversible step of glycolysis. There are three irreversible steps of glycolysis. Those are hexokinase glucokinase enzyme involved in step 1 of glycolysis, where the phosphorylation of glucose is occurring. Next is phosphofructokinase involved in step 3 of the glycolysis. So after that, which is also rate limiting enzyme of glycolysis, pyruvate kinase, which we just now discussed, pyruvate kinase, which is also the irreversible step of glycolysis, step 10 of glycolysis. So there is another enzyme ending with the word kinase, phosphoglycerate kinase, but this enzyme is not in the step 7. This enzyme is not involved in irreversible step of glycolysis. Okay, so why we need to remember the irreversible enzymes that are involved in irreversible steps of glycolysis? Because we need to understand about the regulation of glycolysis. So these are only the enzymes that are involved in the regulation of glycolysis. Okay, so let's look on to the consumption of ATP in glycolysis. So actually 2 ATP has been consumed in the initial stage of glycolysis. So in step 1, the enzyme involved is hexokinase glucokinase. So this will phosphorylate the glucose. So during this process, 1 ATP is consumed, another ATP is consumed in step 3 of glycolysis, where the enzyme involved is phosphofructokinase, where the fructose 6-phosphate is again phosphorylated to form a fructose 1,6-bisphosphate. So total of 2 ATP has been consumed in the energy investment phase of glycolysis. So now energy production in step 6 and step 6, glycerol dihydrophosphate phosphate dehydrogenase enzyme will release NADH during the process, where glycerol dihydrophosphate is phosphorylated to form 1,3-bisphosphoglycerate and oxidize also. Okay, so this NADH will enter into electron transport chain through malate aspartate enter into the mitochondria through malate aspartate shuttle and participate in electron transport chain, and by the process of oxidative phosphorylation it will produce around 3 or 2.5 of ATP. 3 ATP is produced as per the older calculation, and as per the new calculation it produces around 2.5 of ATP. So total of 2 NADH will be produced from the 6 carbon glucose to form a 6 or 5 ATP. So total 6 or 5. Okay, so step 7 is an example of substrate level phosphorylation, where the enzyme involved is phosphoglycerate kinase, where it releases phosphate from 1,3-bisphosphoglycerate to 3-phosphoglycerate. So this is an example of substrate level phosphorylation, where 1 ATP, or when we talk about both the 3 carbon compound of glucose, then total of 2 ATP will be consumed in this reaction. So in the step 10, the enzyme involved is pyruvate kinase. Phosphoenol pyruvate is now releasing its phosphate to form a 3 carbon compound pyruvate. So during this process, ATP has been formed, and this is also another example of substrate level phosphorylation. So 1 ATP or total of 2 ATP will be produced from this step. So let's look at this table to understand the production of ATP in glycolysis. These are the list of enzymes involved. So hexokinase, glucokinase, there is a consumption of ATP. So phosphofructokinase also consumption of ATP. So now glycerol dihy 3-phosphate dehydrogenase will produce NADH through oxidative phosphorylation. According to the new calculation, it will produce total of 5 ATP, and according to old calculation, it will produce total of 6 ATP. Another is phosphoglycerate kinase involved in a step 7 of glycolysis, involved in step 6, step 3, step 1. Okay, so in step 7 of glycolysis, phosphoglycerate kinase enzyme is involved, so it produces ATP. It produces around 2 ATP, no matter old or new calculation. So next example of substrate level phosphorylation is from the step 10 of glycolysis, that is pyruvate kinase. Okay, so it will also produce 2 ATP. So total, if we calculate 5, 6, 7, 8, 9, there will be a total of 9 ATP produced as per the new calculation, but we have 2 ATP to pay back the loan. Okay, so that's why we will have total of 8 ATP, 7 ATP. As per the old calculation, we will have total of 10 ATP, so we need to pay back the 2 ATP, so we will have total of 8 ATP, net 8 ATP from as per the old calculation. So which enzymes are inhibited in the glycolysis? In step 6, glycerol dihy 3-phosphate dehydrogenase enzyme is inhibited by iodoacetate and arsenate having a 5 valency, and step 9, the enzyme enolase is inhibited by the compound fluoride. Okay, so regulation of glycolysis, we have already studied the irreversible step of glycolysis. So these 3 enzymes involved in irreversible step of glycolysis actually regulate glycolysis. So those are hexokinase, glucokinase, phosphofructokinase, and pyruvate kinase. Okay, so the hexokinase is inhibited by its own product, glucose 6-phosphate, whereas glucokinase is not inhibited by its product, glucose 6-phosphate, but glucokinase is induced by insulin, whereas glucohexokinase is not directly induced by insulin. Another enzyme involved in irreversible step is phosphofructokinase. This is also considered as a rate limiting enzyme. So it's allosteric inhibitor. This is inhibited when there is a presence of ATP, citrate or citrate is also a potent inhibitor of glycolysis and hydrogen ion or low pH. In that condition, phosphofructokinase is inhibited, whereas phosphofructokinase is activated when there is a presence of fructose 2,6-bisphosphate, adenosine diphosphate, adenosine monophosphate, or inorganic phosphate. So allosteric activator, fructose 6-phosphate, we must be wondering, like, where is this fructose 2,6-bisphosphate is coming from? Okay, let me tell you. Fructose 2,6-bisphosphate is formed from the phosphorylation of fructose 6-phosphate, which is produced from the step 2 of glycolysis. This fructose 2,6-bisphosphate is formed by the phosphofructokinase 2 enzyme. Okay, it's a bifunctional enzyme. Phosphofructokinase 2 will form a fructose 2,6-bisphosphate. Again, its counterpart enzyme, that is fructose 2,6-bisphosphate, will degrade the fructose 2,6-bisphosphate. So they comes together. Phosphofructokinase 2 and fructose 2,6-bisphosphate enzyme is a bifunctional enzyme. They both comes together. So fructose 2,6-bisphosphate is produced from the phosphofructokinase 2 enzyme. Okay, which will activate the glycolysis reaction or activate the glycolysis reaction. So next is pyruvate kinase. It is also pyruvate kinase is inhibited by accumulation of ATP, whereas it is activated when there is an accumulation of fructose 1,6-bisphosphate, which is produced in the step 3 of the glycolysis. Okay. Pyruvate kinase is active in dephosphorylated state and inactive in phosphorylated state. Inactivation of pyruvate kinase by phosphorylation is brought about by cyclic MP dependent protein kinase. So that was all about the glycolysis details part 2. If you have any question, then please mention it in a comment box. Constructive and positive feedback are welcome. If you like how I explain the topic, or if you think it's valuable, then please do subscribe my channel, like and share it with your friends as well. See you again in a next topic. Okay.